In this lecture, I am going to discuss DNA structure. Main points of the lesson are double helix, nucleotide, purine and pyrimidine bases, directionality 5' to 3', phosphodiester linkage, major and minor groove. In eukaryotes such as plants and animals, DNA is found in the nucleus, a specialized membrane bound compartment in the cell. DNA is also present in certain other types of organelles such as mitochondria and chloroplasts of plants. In prokaryotes such as bacteria, DNA is not enclosed in a membranous envelope, although it is located in a specialized cell region that is called nucleoid. The three-dimensional double helix structure of DNA correctly explained in detail by James Watson and Francis Crick. Inside the eukaryotic cell, you can find double-stranded DNA in chromosome. The two strands intertwine to form a double helix. DNA molecule has two strands and each strand is a polynucleotide chain. Means each strand is made up of many individual units called nucleotides. What does a nucleotide look like? This is a nucleotide. DNA building block is a nucleotide. A nucleotide has three components. Number one is pentose sugar, that is called deoxyribose. DNA contains the sugar deoxyribose while RNA contains sugar that is called ribose. The only difference between ribose and deoxyribose is that ribose has hydroxyl group attached to second prime carbon and deoxyribose has hydrogen atom that is attached to second prime carbon in the ring. Second component of nucleotide is a phosphate group which is attached to 5 prime carbon, and third component is one of four possible nitrogenous bases, that is adenine, guanine, cytosine and thymine is attached to first prime carbon of deoxyribose. So, this is deoxyribose and at first prime carbon one of the nitrogenous base is attached and with fifth prime carbon a phosphate group is attached. It is conventional to number the carbon atoms in a molecule using a system devised by organic chemists. Accordingly, in nucleic acid chemistry, the individual carbon atoms in each sugar and in each nitrogenous base are numbered. The carbon atoms in a base are designated by numerals like 1, 2, 3, 4, 5 and so on. But the carbon atoms in a sugar are numbered as first prime, second prime, third prime, fourth prime and fifth prime. Proceeding clockwise from oxygen atom, a nitrogenous base is attached to first prime carbon and phosphate group is attached to fifth prime carbon. In addition, a free hydroxyl group is attached to third prime carbon. There are two kinds of nitrogen containing bases, that is purines and pyrimidines. Cytosine and thymine are called pyrimidine, contains only one ring of atoms, whereas guanine and adenine are called purine, which contains two rings of atoms. The name of nucleotide depends on the presence of nitrogenous base. Like thymine nitrogenous base is bonded to deoxyribose sugar, so it is thymine nucleotide. Here adenine nitrogenous base is bonded to deoxyribose sugar, so it is adenine nucleotide. Here cytosine nitrogenous base is bonded to deoxyribose, so it is cytosine nucleotide. And here guanine nitrogenous base is bonded to deoxyribose, so it is guanine nucleotide. The nucleotides like guanine nucleotide, cytosine nucleotide, adenine nucleotide and thymine nucleotides are linked by covalent bonds to form an alternating sugar phosphate backbone. The backbone of each strand is made of alternating sugar, deoxyribose and phosphate group. What is a phosphodiester linkage? Nucleotides like here guanine nucleotide and cytosine nucleotide, they are linked together by this linkage which is called phosphodiester linkage. It is a dehydration reaction involving the 5' phosphate. This is 5' phosphate because it is attached to 5' deoxyribose sugar. So, this 5' phosphate of one nucleotide is bonded with 3' hydroxyl of another nucleotide. This linkage is called phosphodiester linkage because the phosphate group is now linked to two sugars by means of a pair of ester bonds. So, one end of phosphate group is attached to 5' carbon and the other end of phosphate group is attached to 3' hydroxyl of deoxyribose. So, phosphodiester bond is the bond which occurs when phosphate it forms two ester bonds, and many thousands of nucleotides can join together via these linkages to form a long chain nucleic acid polymer. So, here phosphate group is already attached to 5' carbon of deoxyribose, and this linkage between phosphate group and 5' carbon is phosphoester bond. This is phosphoester linkage. And of course, this phosphate is also attached to 3' carbon of deoxyribose by another phosphoester linkage. So, together two phosphoester linkages, this is one ester bond and this is second ester bond, together it forms a phosphodiester bond, that is also known as phosphodiester linkage. Linear strands of DNA or RNA, no matter how long, almost always have a 5' phosphate group at one end and 3' hydroxyl group at other end. So, the carbon atom in this key describes the directionality of DNA strand is 5' to 3'. Now, looking within the sugars, there is intrinsic orientation difference between the two strands of DNA. Proceeding clockwise from oxygen atom, this is first prime carbon, second prime carbon, third prime carbon, fourth prime carbon and fifth prime carbon, and phosphate group is attached to fifth prime carbon. So, on the top strand you can see 5' carbon on each sugar is on the right and 3' carbon is on the left. So, the direction of top strand is from right to left. Now, proceeding clockwise from the oxygen atom on the bottom strand, you can see this is first prime carbon, second prime carbon, third prime carbon, fourth prime carbon and fifth prime carbon. So, fifth prime carbon is on the left and third prime carbon is on the right. So, the direction of bottom strand is from left to right. So, the top strand orientation is from 5' to 3', that is from right to left. So, the two strands are anti-parallel and sometimes called Watson and Crick. The polarity or directionality of polynucleotide chain with 5' at the top and 3' at the bottom. So, each polynucleotide chain is directional and it is directed from 5' to 3'. The two strands, they interact through non-covalent hydrogen bonding between the bases. Further examination of the model indicate that adenine nucleotide can pair with thymine and guanine is paired with cytosine in such a way that hydrogen bonds form between them. There are 3 hydrogen bonds between guanine and cytosine and 2 hydrogen bonds between adenine and thymine. Hydrogen bonding, it contributes the specificity of base pairing. This concept of base pairing neatly explains Chargaff's rule. The amount of cytosine must equal to the amount of guanine because every cytosine in one chain must have a paired guanine in the other chain. Similarly, every adenine in the first chain must have a thymine in the second chain. So, the sequence of bases of two chains, they exhibit complementary base pairing. That is, the sequence of nucleotides in one chain dictates the complementary sequence of nucleotides in the other chain. For example, if one strand has the sequence of adenine, guanine, cytosine, thymine, adenine and cytosine, the other strand has complementary sequence of thymine, cytosine, guanine, adenine, thymine and guanine, because adenine is paired with thymine and guanine is paired with cytosine. DNA has a regular structure. Its orientation, width between the nucleotides, number of nucleotides per helical turn is constant. Each pair of bases is exactly 0.34 nanometer from adjacent pairs above and below, because exactly 10 nucleotides are present in each full turn of helix, and each turn it constitutes 3.4 nanometer of length. As we know, the two chains run in opposite direction. Therefore, each end of double helix must have an exposed 5' phosphate in one strand and exposed 3' hydroxyl group on the other chain, because the two strands, they are running in opposite direction, so they are anti-parallel. Cytosine and thymine are pyrimidines, contain only one ring of atoms, whereas guanine and adenine are called purines, which contains two rings of atoms. If each rung of the ladder contained one purine and one pyrimidine, the width of helix at each base pair would be exactly 2 nanometer. What are major and minor grooves? DNA has two types of grooves, that is minor groove and major groove. Minor and major grooves, they are opposite to each other and each runs continuously along the entire length of the molecule. The major groove occurs where the backbones are far apart, and minor groove occurs where the backbones are close together. Major and minor grooves arise from the anti-parallel arrangement of two backbone strands. The grooves are actual structural features of the molecules, and the grooves are important in the attachment of DNA binding proteins involved in replication and transcription. Replication means copying a DNA to DNA, and transcription means copying DNA to RNA.
Structure of DNA. The DNA molecule is a polymer consisting of several thousand pairs of nucleotide monomers. Each nucleotide consists of three components. They are 1. Pentose sugar, deoxyribose, 2. Phosphate group, 3. Nitrogenous base. Nucleoside means deoxyribose plus nitrogenous base.